Greetings to everybody from Mexico. Namaste. Okay, in next few minutes, I will talk about my work on gene editing. And let me share my screen. Dr. Bhatt, can you see my screen?
Yeah, perfect. Please go ahead.
Gene editing is such a, let me switch off my camera maybe so that we can have better bandwidth. Okay, I think it's okay. So gene editing is such a powerful and versatile tool that it can seamlessly integrate.
Excuse me, doctor, can you put in the slide? Yeah, slide view. Yeah, it's coming now. Fine, perfect.
Sorry. Gene editing is such a powerful and versatile tool that it can seamlessly integrate any crop breeding program. So be it rice, wheat, maize, or even any spices. In next few minutes, I will walk you through why gene editing is important for crop breeders, how we have successfully used it to reduce the stomatal density in rice, and our progress in wheat and maize work. Of course, I did not keep maize by keeping the time limit in mind, but we can discuss something on that. And if time permits, I will also discuss about some recent improvements to CRISPR-Cas system and the regulatory benefits that the CRISPR crops can enjoy. The world population is growing continuously, and whatever the land, there is not much change in the arable land from 1965 till today. But if you compare the per capita land availability, it has become less than half. And this is not true. If we go in real terms, actually a major part of this is going for food and going for the industrial production of raw materials and also for biofuel. So we have to feed the population with this available land. The world population got doubled between 1960 and 2000. And thanks to Green Revolution, which gave a new thrust to crop productivity to support the cereal growth mainly, and that has supported the population blast. Though this has very successfully maintained the sustainability of crop productivity till year 2000, but now the crop productivity is going to face new challenges. For example, there is several new strains of pathogens have been recently discovered. For example, there is one strain called UG99 for wheat rust that appeared in 1998, and within few years it has spread to 13 different countries. And most of the resistant varieties that we have previously CIMMYT had released, it has broken down all the resistance. I'll come a little bit more detail into this why it happened later. But there are some other things like spread of pathogens to different horizons. For example, on the right side, you can see the yellow rust, which was usually adapted to a warmer climate. Now it is spreading throughout the world. The effect of climate change, everybody is aware. And one of the side effects of the Green Revolution was that it has narrowed the genetic diversity. And above this, the poor combining ability of the resistant varieties with the elite lines poses a new challenge for the breeders. Genetic engineering offered a very precise and exciting opportunity to transfer the traits across the genetic barrier. But unfortunately, the use of the markers that the scientists use to develop transgenic plants, that has created a widespread speculation among the consumers that they may develop drug resistance. In spite of that, we have great success in herbicide-tolerant corn and BT cotton of India. And today, I was just reading a report by ISA that the cultivation of genetic crops has crossed 1 billion hectares. But in spite of that, there are many countries like European Union, say India, and who say big no-no to transgenic food crops. Therefore, the crop biotechnologists have always been looking for a model that combines the best features of transgenic technology with those of organic and conventional farming. And the site-specific nucleases offer exactly such a solution. Let me show you why site-specific nucleases are so important and how they can help in overcoming the difficulties of transgenic technology. So this can be compared with our day-to-day cut and paste in Microsoft Word or in our cell phones. So this technology is not new. It has been there for quite a long time. But before 2013, most of the gene editing was done using zinc finger nuclease or by TALEN. Unfortunately, both of the technique needs, like zinc finger nuclease needs involvement of protein engineering for DNA protein binding interaction. And similarly, the TALEN, though it is a little bit easier, the protocol is very difficult because the construct is generally unstable. So that's why people are looking for some alternative, and it was limited to only specialized labs. So when the CRISPR was introduced in 2013, it kind of revolutionized everything because here we have to design only a very small piece of guide RNA that is about 20 nucleotides. And that's all that we have to provide to guide this Cas9 enzyme to the target site. So this technology, the delivery of the tools exactly works like transgenic technology. You have a guide RNA, you have a Cas9 enzyme, you have a guide RNA gene, and these two have to be combined with a marker gene like transgenic technology so that it can go and we can select the transformed plants. This can be transformed using biologistic gene gun method, or you can use agrobacterium mediated transformation. And once this has been integrated into the genome, that is very random kind of integration, the Cas9 gene starts expressing and the guide RNA guides the Cas9 to the target site. Once the target site is recognized, the Cas9 makes the change there. And remember, we have just delivered the tool to make the change, but not the change itself. So once the change is done, these tools can be segregated. These tools can be segregated out, and we can get a plant absolutely free of any transgenic traces. Now that we have an idea about the technology, let's see how it is going to help in breeding pipeline. So once breeders identify a QTL or a gene, which usually takes several years of effort, but once they identify, they try to cross it with an elite line to introgress the gene of interest into the elite line. But since this is not very precise, there can be integration of few other genes which are not agronomically wanted. So basically, they can be something like scattering of seeds or some other traits, or drooping leaves, which the farmer doesn't want. So for that, the breeder has to do several rounds of marker step backcrossing to get a plant which is near isogenic to the elite line, elite parent. With the development of technology, with markers, with genomic markers, all these things, still it takes 7 to 12 years to develop and release a variety. Instead, this can be done just within 2 to 3 years by using homology directed recombination or by prime editing. Those are different terms for CRISPR-mediated gene editing. I think we don't have scope to discuss all those, but basically these are simple methods that gene editing can use to deliver a gene fragment into the plant. And there is no linkage drag, so this can be achieved much faster. And this is not, I'm not just telling some story. Already the maize, gene edited maize that has been released, that was done in 42 months. So that's the proof that we can do it within 2 to 3 years. With this, I will take you to some of my work that we did in rice. So when we were starting this work in 2013, that time CRISPR technology was very new. And we wanted to evaluate this technology and also to get some benefit of this technology. So we targeted a gene called epidermal patterning factor like 9 or APFL9 signaling peptide gene. So this signaling peptide, it's also called stomagen. It acts on a series of genes finally to develop the stomata or guard cells. And we selected this gene because it was known in Arabidopsis that when this gene is overexpressed, it increases the number of stomata. And when it is silenced, it decreases the number of stomata. And this gene also expresses in the mesophyll cells of Arabidopsis. So we studied this and we wanted to knock out this gene in rice and see the effect. That time, in addition to Cas9, there was a new CRISPR factor was developed called CPF1 or Cas12A. So the difference between these two is the Cas9 enzyme, which is very standard enzyme that is used mostly, that needs a, in addition to the spacer, it needs a small element called protospacer adjacent motif, which is generally NGG, means N is any nucleotide, and after that there should be GG. So that puts a restriction that your genes should have this near the spacer. Sometimes for some of the genes we don't get this. This CPF1 instead it uses a PAM, which is TTTN. So basically any gene which if it is AT rich, you can use this. So we wanted to validate both genes, and this is the EPFL9 gene. We targeted the first exon so that the complete gene is knocked out. And we used very close targets for both Cas9 and CPF so that we can compare the effect. And these are the guide. This is the guide RNA. This is the PAM. This is guide RNA for CPF, and this is guide RNA for the PAM. So this is PAM for the CPF. We developed about 100 plants, 100 independent events for both constructs, and then we evaluated these mutants by an enzyme called by a nuclease called surveyor nuclease. This is a very simple protocol, and we use this because we don't need any prior knowledge of the kind of mutation that we are going to have. So all that you need is have an idea about the target site. So if you have an idea about the target site, you amplify a DNA fragment flanking the target site, both from wild type and also from the expected muted plant, expected mutant. And you just hit nature and just try to re-anneal. So there can be 3 kinds of re-annealing. Wild type can anneal with wild type, mutant can anneal with mutant, and also there is a chance that wild type can anneal with mutant. If this happens, so this is the target of this surveyor enzyme. If there is a mismatch, it makes a double-stranded cut. And in that case, we can get 2 different bands of DNA. So these 2 generally will be of almost same size because there will be few bases addition or deletion that will not make much difference in the gel, but these 2 will give 2 clear bands. And we tested our plants. We saw both Cas9 and CPF generated plants had mutations, and then we sequenced these using we just PCR amplified and sequenced using Sanger sequencing. And as you can see, there was mostly it was deletion. We had no insertion in this case, but insertion do happen. So we got mostly deletions here in Cas9. Also, same thing we saw in CPF. For Cas9, we got something from 4 to 37 base pair deletions, but for CPF, we got from 1 to 64 base pair deletions. With this, when we translate these deletions, as you can see, suppose there is one base deletion, that what happens if it is a non-triplet? If it is a triplet, you can get one amino acid added or deleted. But if it is non-triplet, then the whole coding sequence changes, and that's called a frame shift mutation, and the whole protein gets changed. And if you have started well in the first exon, that means you have hardly anything left from the original protein. And most importantly, you are going to get some kind of stop codon downstream of the target site. So we tried to see the inheritance pattern of the mutations. We took two events. Was having 4 base pair deletion. We tried to grow few plants, and you can see it had a Mendelian segregation in T1, but in T2, when we moved the homozygous plants, all of the plants were mutants in the T2 generation. Then we checked the stomatal, we counted the stomatal density, and you can see in this picture, there was at least about 8-fold decrease in the stomatal density. At this point, I left Erie and I joined UNC North Carolina. So this work is still being carried out for the evaluating about the water use efficiency and if this can be deployed for the drought tolerance. So I'll stop this work here. Then I'm going to, this work was published also in Plant Cell Report in 2017. Then I'll take you to my current work at CIMMYT. I joined CIMMYT about 1.5 years before, and I started working in two main crops, maize and wheat. And today I will discuss mostly about my wheat work. So there are many major diseases of wheat, and it has been shown that about 18% of the crop loss is mainly because of different pathogens and diseases. So in addition to that, there is a secondary effect that these pathogens, they create some toxic products like aflatoxin that is toxic both for human and cattle. So that's why I have focused, I have decided to use the CRISPR technology to improve the disease resistance. This anneals with this kind of aligns with my PhD work where I started with BT rice. And here I'm focusing on wheat rust, powdery mildew, wheat blast, and fusarium head blight. I will discuss about the progress about first two diseases. So rust, wheat rust is very widely spread, and there are three kind of rust diseases: leaf rust, stripe rust, and yellow rust. So this is leaf rust. You can see the whole field, and even the spikes can be affected. This is the yellow rust. The whole crop can be affected, and this is the stem rust. And at worst conditions, you can get 100% yield loss, and you can get no seeds. This disease is, this slide is, the colors doesn't mean much except they show the type of rust. So, but the take home message from this slide is this disease is omnipresent in all continents, and the annual yield loss can be up to $5 billion US dollars. There are more than 200 genes have been discovered for wheat rust, different kind of wheat rust. And mainly these genes can be divided into two subcategories. The single origin mediated resistance that depend upon the interaction between gene to gene interaction between the host and pathogens. The pathogens have a kind of gene called avirulence genes, and these genes interact with that to give a give resistance. In case there is a mutation in the pathogen AVR gene, so these genes will fail. And that's why when these R genes are deployed singly, many cases they have been easily taken over by the pathogens. For example, CIMMYT had deployed so many kind of wheat varieties in Africa. When Eugene 99 appeared in 99, all the genes were broken down. All the resistance genes were broken down, and there was almost zero harvesting in wheat. Good thing is these genes work through all growth stages. That's why CIMMYT breeders have been trying to combine multiple R genes to develop resistant lines. And a new approach that CIMMYT breeders have been taking is using the susceptibility factors. So these genes, like AVR genes in pathogens, these genes work in the plant and they help the pathogen to grow. But in case there is a mutation in these susceptibility factors, then the pathogen cannot grow. Unfortunately, they don't give full resistance, they give only partial resistance. But the good thing is they work over many kind of pathogens and many races. And these genes work only in adult plants, they don't work in seedlings. There are mainly 4 of this kind of genes have been discovered, which give very wide and effective resistance, though partial. LR34 has been there since I think so many years, I don't know. LR46 is there for at least 50 years. LR67 is a new addition. So LR67 is a gene that gives resistance to leaf rust, stripe rust, yellow rust, powdery mildew. So basically all 3 rust and also powdery mildew. It doesn't give 100% resistance. As you can see here also, there is quite growth of the fungus. This is the leaf, this is powdery mildew. So they don't give 100% protection, but they give an effective level of protection so that farmer can get a good harvest. There is not much difference between the resistant and the susceptible protein. There is only one amino acid difference here and one amino acid here. And it has been shown that when they change this amino acid of the resistant variety to the susceptible type, arginine to glycine, that changes the sugar transporter. Sorry, I forgot to say that this LR67 is a sugar transporter gene that helps that helps in transporting sugar from the plant cell to the apoplast, where the fungus can take the sugar using its IV. And this sugar transport is disturbed by this mutation. And when we change back from arginine to glycine, again it starts. But this has the second one at 387 amino acid doesn't have much impact. We also tried to model this these 2 proteins, resistant and susceptible, and you can see that the resistant variety forms a alpha pi helix here, and that way it forms a constriction in the sugar transporter, and the sugar transporter fails to transport the sugar. So this can be with this information, we are trying to knock out this gene. So we could have modified exactly that amino acid using base editing, but these eukaryotic plants, they have many sugar transporters, and if you knock out one of these, there won't be much impact for the plant. Whereas these pathogens, fungus, they're very specific for the specific kind of protein or transporter, so they don't usually grow. That has been proved in many crops. That's why we are trying to knock out this gene from this fast exon. So unfortunately, this wheat is a allo hexaploid crop. There will be 3 copies of 3 alleles of each gene. So we have to knock out all 3 genes to get durable resistance. And that is another reason, I'll show in few other slides, that why this is, why wheat will love the genome editing than the breeders' efforts. So wheat has remained as an orphan crop from long time. Unlike rice and maize, where the transgenic has been developed long back, like rice, I don't know from what time. I think 2004, we had the best protocol, which works seamlessly. Like you transform 200 cali and you get enough number of plants. And maize, the first maize transgenic crop was released already in 1996. But unfortunately, wheat has remained a orphan crop and not much has been done. So it took us almost a year to standardize a good protocol for wheat transformation. But now we are happy that we have a very effective protocol. We can develop transgenics for any wheat elite lines. Like we are not working with only just with the test lines. We are working with the elite line called Riedling and Baz, Kachu. So these can be directly given to the farmers. So we have developed a very nice protocol, and we have got now transgenic plants, and we are now evaluating this for the gene editing and also for their resistance to different kind of wheat loss. The another disease that I'm interested and we are working is called powdery mildew. This is also widespread in India, China, and many other countries. And the yield loss can be as high as 40, it's generally about 40% yield loss, and it can be as high as 100% also. And some work has been done on these before. When these polyploid crops are dealt with breeders or pathologists, they try to find some mutants, and it is very, very difficult to find mutants for all 3 genomes. Like wheat has A genome, BB, and DD genome. So it's a allohexaploid, and it is very difficult for breeders to get mutation in all 3. That's why the pictures that I showed you, which were collected by our wheat breeders and pathologists, they never show 100% resistance. So this work was done in China, and they tried to knock out individual genes AA, BB, DD, ABB, ADD, and all 3. And when knocked out all 3, then only they got very high level of resistance. And we have also, you can see here, almost no infection. So this is the MLO gene. This was initially discovered in barley, and we have 3 orthologs of that. But the good thing is, the genes are about 90–80% similar in sequence, and it's very easy to find a target which is similar in all 3 genes. So with sequence alignment, we have identified this region. This will make a cut here, and we have also transformed, and we have got transgenic plants that we're evaluating now. So that's kind of end to my current work. I see it's already 27 minutes, and I think I'll take another 3–4 minutes to discuss about some exciting improvement to this technology. Hopefully I have that much time. So I showed you in our LR67 work, we are trying to knock out the gene at 2 different places. So because sometimes if the gene is present, that can create some kind of regulatory inhibition, even if it is a non-functional protein, and even if it is, we know that it gets degraded very fast by a mechanism called non-specific degradation of proteins. But it can still regulate, it can still attract some regulatory stringency. For that reason, we are trying to knock out the complete gene by cutting at both ends. This technology was developed in University of Pennsylvania in US. So usually the tRNA is processed by two enzymes after its initial production. And these two enzymes that cut at both ends of the tRNA to release the tRNA. And in case if we can add our gRNA in between two tRNA sequence, it can be automatically processed by the cellular system to release our guide RNA. And we use this technology to, I have been using this for quite a long time, and I have been able to generate triple mutants very shortly within 6 months time. And this can be used for even up to 8 targeted mutations. But one negative thing about this is when you make multiple cuts, that can lead to rearrangement of the genetic fragments. So here, the base editing is another technique that gives the mutation without any double-strand break. And most of the diseases that we know so far are because of single nucleotide polymorphism. So when we add a deaminase enzyme to the Cas9, in an inactivated Cas9 protein, that can be targeted to the site of modification, where the cytosine can be modified to uridine, which can be later changed to thymine. And when the uridine is changed to, when the cytosine is changed to thymine, the other strand can be changed to from guanine to adenine to match it. Of course, the technique is not that simple, because sometimes if this uridine is getting introduced, that can be removed by the cell to repair it. But we have some other things, like we can add some UGI, uridine glycosylase inhibitor, and some other things that can make this change stable, and so that we can get change from GC to AT. Similarly, adenine base editors have been predicted, and in both ways, we can change today any nucleotide you say, we can precisely change it to get back or repair the gene. So this has been used in rice already to change a rice blast resistance gene, and they have shown that the plants show very high level of resistance without any double-strand break. So this can be very easily compared or mimicked to our what our breeders do, or what happens naturally by spontaneous mutation or by chemical mutation. There is another technique called prime editing. So in prime editing, I showed before that if we do homologous recombination, we can repair a gene by replacing with a new copy of the gene. But delivering that homologous copy of the strand is quite difficult and inefficient. Here we can add that template to the guide RNA, and that can be reverse transcribed by reverse transcription enzyme, and that can repair any kind of DNA. So this is the newest addition. And also we can use CRISPR to very precisely detect any pathogen. So this slide seems to be very complicated. Let me take it out. Basically, it depends upon an enzyme called Cas9, which has RNA target. Till now, I have whatever enzyme I have discussed, they target the DNA. This one targets the RNA. So if there is a pathogen which is DNA or RNA pathogen, we can translate the DNA to RNA, or if it is RNA, it can remain RNA. And when this enzyme faces the target, it collaterally clips other RNA molecule with reporter genes that can be detected or even can be visible by naked eye. So if we develop this technique, we can we can enable the farmers, and they can take these strips to the field where they can just take a leaf and they can put a drop of the leaf soup, and they can get to know whether their crop is infected or not. We have initiated a project for the wheat blast for this, and we have not done much progress, but we are just in the stage of working on it. And the last slide is the deployment benefit of of the CRISPR crops, because we don't leave any transgenic trace. So these crops will be similar to the crops that is generated by breeders by spontaneous mutation or by chemical mutation. So USDA has already approved the CRISPR-edited mushroom and CRISPR-edited maize, and they say that there will be no regulation on the plants, CRISPR-edited plants, if that could otherwise have been developed by conventional breeding techniques. So the plants that I discussed about LR67 or MLO, since those are already present in the nature, we expect there will be no regulation. And luckily, there are some good people still today. So we have got a license for we have got a philanthropic license for the Cas9, and we expect that we can release our wheat plants very soon. I did not include my maize work here because of the time constraint, but I'm happy to say that we are negotiating. We have already got gene edited plants. We are negotiating with the government of Kenya and Kenya and a few other countries for our field testing of gene edited maize lines there. Other countries like Israel, Brazil, Argentina, Colombia, Paraguay, and Chile, all of them have similar approach. If there is one or few change, they will not regulate the plant. India, there is a draft proposal that they will consider case-to-case basis. But I hope if it is one or few change, they will also not regulate it. With this, I thank you, Dr. Bhatt, and the team and all the participants for listening, for your careful listening. And I'll be happy if there is any question to take. Thank you.